Hello, we are Senior Design Team 1 from the Biomedical Engineering Department. Our project was to create and test a new blubber-only implantable satellite telemetry tag. We were sponsored by Dr. Rajachar in the Biomedical Engineering Department. With many species becoming critically endangered, understanding their movements and habitat use are critical for conservation efforts. The use of satellite telemetry tags allows for these movements to be recorded. The problem with current tags is that they do not last in the animal for an entire migration cycle. This makes it difficult to create protected areas which reduce the amount of human activity that may cause harm to the animal's population. The team's objectives were to create a new tag design that incorporated new retention elements, as well as develop a consistent testing protocol. The team underwent a design process which evaluated the addition of macro features and micro features to increase retention of shortened tags within the blubber that lead to the decision of a new macro feature. The prototype tags that will be tested are a featureless control tag, a cup circle tag that will be deployed this fall, and an inverted screw tag, where threads would switch directions every third of the way down the shaft. These will be tested for penetration depth, pull-out force, performance changes due to a microfeature, and tissue damage. A small-scale testing machine was then created to accurately mimic the tagging tools used in real-life applications. This machine was used to evaluate how much pressure is required for the tags to penetrate into a blubber sample at an optimal depth.
Penetration was tested by placing the small-scale prototypes of the tags into the testing machine right below the pressure valve. Each tag was tested at a constant 40 psi into a small piece of whale blubber with the skin attached. These samples were then transferred over to pull-out testing. Our results showed that the penetration depth is directly proportional to the size of the macro feature on the tag. The larger the surface feature, the more pressure is required to reach optimum depth. Pull-out testing was done to measure the force displacement curves of each tag after being fully removed from the blubber. This data was found to be inconsistent due to an overhang in tag tip. These tips were filed down and retested. The average maximum force along with the average energy was then calculated and compared.
After comparing the pull-out data for the three tag designs, the cup circle design required significantly more force to be removed from the blubber than our team's inverted screw design. However, the two tags had similar average energies to remove the entire tag. It was also seen that the amount of force required for retention significantly decreased after the tag tips were filed down. The addition of a micro feature was then analyzed by sandblasting the top 1/3 of the shaft of a featureless control tag. The tag was set inside a gelatin solution overnight. The tags were then pulled out of the gelatin the next day. Sadly, the RPM value for the sanded portion was unknown. The addition of the sandblasting showed a decrease in the maximum force. This means that the sandblasting process should be set at a higher RPM to make the tag rougher to cause a more desired result. However, the average energies of the control tag and sanded tag were extremely similar, and there were no significant differences in the data sets.
We determined that the tag tips being filed down to be in line with the tag body drastically reduced retention. After this, we confirmed the cup circle achieved maximum retention properties for surface features. The team developed a new inverted screw retention feature and established more repeatable penetration testing protocol. Due to the reduction in time to work on the project from COVID-19 outbreak, we were unable to continue testing the effects of surface roughness on retention. Tissue damage was also unable to be reviewed, and optimization of the new inverted screw design to enhance its retention properties. We would like to thank the following people on this lab.